Hi, welcome to Microbiology and Parasitology Laboratory. This is exercise 1, Aseptic Techniques. Our knowledge of microbiology has come through the study of isolated species growing in an environment free from contamination by other living forms. Further advances in the different areas of microbiology will depend upon studies of individual microorganisms in pure culture. Pure culture techniques have resulted in the accurate characterization and identification of most microorganisms. Since most microbiological studies utilize pure cultures, outside contamination should be avoided. Procedures which prevent contamination by unwanted microorganisms are commonly referred to as aseptic techniques. Let us discuss some of the general rules relating to good aseptic techniques. First, we have the hand washing. Whether you wear your gloves or not, you should always wash your hands thoroughly before and after your work or whenever there is a possibility of contamination. Disinfect the bench top before and after working in the laboratory. Commonly used disinfectants are 10% bleach or sodium hypochlorite and ethanol. These are recommended because of its rapid action that usually takes around 5 minutes contact time. While working, observe the general rules for laboratory safety. So you should be aware of the hazards that are present in your work area. So when you are working with an open flame or volatile and flammable liquids, and you should also know the location of emergency exits, emergency showers, and others. For any new procedure, identify those points where the control of microbes is most critical. Now, if you have some questions or you need some clarifications, do not hesitate to consult your laboratory preceptor. Rules for using and sterilizing inoculating loops and needles. When you are working with inoculating loop and needle, it should never be let out of your hand from the time of sterilization through the time of transfer and until the time of inoculation. There are different types of inoculating loops and needles. Some are reusable, those are made up of nichrome wire, while some are disposable, which are made up of plastics. Let us proceed to the sterilization procedures. So first is you hold your wire loop or needle like a pen. So in this way, it leaves your little finger free to hold your screw cap or tube cover or let's say cotton plugs while working. Now you hit both the loop or needle and the lower part of the handle over the Bunsen burner flame. So when we are working with Bunsen burner, remember we always use the blue flame. The Bunsen burner flame also creates an air convection current which keeps away any organisms that is suspended in dust particles. So it somehow provides protection or a shield within your immediate working area. For faster heating, hold the loop almost vertically. Holding your loop at an angle almost vertical allows any liquid culture to flow into the flame. Now, using Bunsen burner is not always practical in all situations. The Bunsen burner or the use of open flames is not allowed when working under a biosafety cabinet because it causes disruption in the air curtain within your biosafety cabinet. As an alternative, other laboratories are using microincinerator. So for the heating of your loops and needles, we begin at the junction of the wire and the handle. So this is where is it located, and then you slowly moves towards the loop end as the wire glows red. So heating your loop or needle to red hot ensures that bacterial spores are destroyed. If a loop holding organisms is thrust into the flame, the rapid evaporation of the moisture creates an aerosol, and this can result in a dangerous spray of microorganisms. Ensure that the entire wire portion of your loop is heated prior to use and again immediately after use. This is a demonstration of how is it performed. Remember, we use the blue flame and starts at the junction of the wire and the handle, slowly heating it to red hot as you move towards the loop end. After sterilization, allow it to cool and do not let it come in contact with anything. Now we have the rules for handling culture plates. Uninoculated plate that is accidentally touched or dropped should be sprayed first with disinfectant before discarding. When streaking plates, minimize the amount of time that the lid is off the plate. Remove the lid of each plate only when you are ready to inoculate. Do not breathe directly into the surface because it might contaminate your culture. All discard plates should be placed in a stainless tray in inverted position to prevent spillage of microorganisms into the environment. These plates are sterilized by autoclaving for 45 to 60 minutes before washing. So there are different types of autoclaves. So this is what we use in the laboratory. Now we have the rules for handling culture tubes. The tubes should be placed in the rack when not in use or whenever you carry them from place to place. They should not be laid on the bench top. An open tube should always be held in hand. The cap or any cover of the tube should never be placed on the bench top. It should be held until the tube is covered and returned to the rack. So that's what I have mentioned earlier. So you hold your tube cover or cotton plugs using your little finger. All discard tubes must be placed in autoclavable containers. These are sterilized prior to washing. For the inoculation procedures, first we have the transferring of slant culture to tube media. So first step is to sterilize your loop or needle. When you are working with slant culture, you may either use an inoculating loop or an inoculating needle. After sterilizing your loop and needle, open your tubes using your little finger and remember, never lay down your plugs. Now, flame the mouth of your tube or slant culture. Flaming the mouth creates an air convection current, which prevents organisms in the environment from entering your tube culture. Therefore, it prevents contamination. Now, you may obtain a small amount of growth from your slant culture, and transfer it to another fresh medium. Now, if you are going to transfer your slant culture to another slant culture, streak it in a zigzag manner. So this is your fresh medium, another slant culture, and you streak it in a zigzag manner. Now, if your medium is a broth, dip the needle or loop in the broth and shake the needle gently to dislodge the inoculum. Now, if you are transferring a slant culture to a stab, this this requires the use of inoculating needle, because if you are going to use your inoculating loop, you will only damage your stab medium. So this is how it is being done. You insert down into the butt of the agar. If more than one tube is to be inoculated, the inoculum may again be obtained from the culture tube before inoculating next tube. Sterilize the needle before getting another inoculum and work as closely as possible near the flame. After that, you may again flame the mouth of your tube and cover it. Then, for the last step, sterilize your inoculating loop star needles. Now let us proceed to the transferring of broad culture to agar plates. So a while ago we are working with slant culture, and now we'll be discussing about broad culture. When working with broad culture tubes, what we use is the inoculating loops. Some of the same procedure applies. Of course, you need to sterilize your inoculating loop, and while holding the sterilized loop, remove the cotton plugs or screw cap from the mouth of your tube containing the inoculum. So this time we have our broad culture. Then again, flame the mouth of your broad culture tube and then insert the sterilized loop to obtain a loopful of your inoculum. While still holding the loop containing the inoculum, again flame the mouth of your tube and return the plug. After returning the plug, return the tube on the test tube rack. Now hold your agar plate with your other hand supporting the bottom using the middle finger and small fingers. Lift the cover using thumb and forefinger to open the agar plate. You may now inoculate the agar plate with your loopful of inoculum following the streaking patterns that will be shown in the following slides. So work as closely as possible near the flame. Now when streaking your plates, ideally you should use sterilized loop for every quadrant. So this will be further discussed in details in one of your future exercises. So others prefer to use a single loop for at least 3 quadrants. So I will show you how is it being done. For example, this is your loop. So you divide your loop into 3 portions. This upper portion of your loop may be used in streaking your first quadrant. So this is how your first quadrant should look like. And then for the second quadrant, you may either use the left or the right side. For example, you may use the left side for streaking your second quadrant. Now you use the opposite side for your third quadrant. So this is your third quadrant. Since the entire portion of your loop has already been used, now you may obtain another sterilized loop to streak your fourth quadrant. So this is your fourth quadrant. That's how transferring of broad culture to agar plate is being done. That's all for exercise 1, and I hope you learned something. Thank you.